Welcome to CHEM104 Lab number 2, Chemical Kinetics. Let's get started with where do you find the material? So first I want to do is share my Blackboard. Where is it? Here. I have to go to there. So when you when you arrive at the course in Blackboard, this is probably what you'll see. Click on Start Here and go to Lab Exercises. And then Chemical Kinetics is lab number 2. What you'll find is the hard copy of the method, Chemical Kinetics student PDF. And let's just take a quick look at that. That's what it'll look like. There we go. There are parts of it in here that I've extracted from the instructor's resource, which I hope will be helpful. And we'll be discussing some of this as we proceed in just a few minutes. There's a lot more of it there. Okay, so that's the hard copy. Then here's the data that you'll need to complete the lab. Right? So this will make more sense as we go along, but you should have everything you need there. We're only going to use the blue dye and combine it with bleach. So we have our 2 reactants, the blue dye and the bleach, for reference purposes in formulating the rate law. So we have to do the experiment, run it once at full strength bleach, which is not really full strength because full strength bleach is 6%. But we're going to use a certain amount of bleach in the first experiment, and then when we go to the second experiment, we're going to use half that strength of bleach. Okay, so there's your data. And the data is in the form of time in seconds, and A, which is absorbance at 630 nanometers. Nanometer is a wavelength. And I've actually left you a column here for the natural log and the inverse. And from our lecture, you already know that this is the straight concentration. This is the log transformation. This is the inverse transformation. And you know, if you graph each of these against time, you'll either get a 0 order, first order, or second order order, yes, for that reactant. And it's up to you to graph each one and determine which one is the proper order for that reactant. In this case, it would be for the blue dye. And later I'm going to show you how to use this information in an Excel spreadsheet to reduce the labor involved. Now, if you really are a hands-on person, you can just do it all by hand. That's fine. It's a little more difficult, but it can be done. Okay, so that's what the data looks like. So let's, and there are a couple other items in here. Well, there's the drop box. So when you finish your report, that's where you drop it. There's also this data in an Excel spreadsheet. Now I'm going to call that up later and show you how that's a sample set, data set. It's not the same data as you're going to be using, but it'll show you how to do these different procedures with the help of Excel. And then here's a document for bleach calculations. So when you're going to need to determine the actual molar concentration of sodium hypochlorite, which is the active ingredient of bleach. You need to determine the actual concentration of sodium hypochlorite in the reaction vessel for the full strength, and then of course just take half of it for the second experiment, 1/2. And later I'll show you what the significance of those values is in terms of developing the rate law. Okay, so there you'll have all your information here that you'll need to complete the report. And now let's get to the let's get back. I'm going to leave that up and just unshare it. Okay, so now I'm going to check things off as I go, so I know I'm got everything, and headed for. Now we're going to talk a little bit about the theory, the kinetics, and this will be something of a review because we've already gone over some of this in the lecture part. But I'm going to try to tailor it to this particular experiment. So when we write a rate law, remember that we have the rate of the reaction is equal to some constant, right? And that's times the concentration of your reactants, and each reactant is raised to a power, which is its order. So in this case, the first reactant will be, let's say we'll make the first reactant the bleach, the sodium hypochlorite. And we'll raise it to the power of M. And then the other reactant is the blue number 1 food coloring. They actually put this stuff in your food. It's food safe. And it's raised to the power of N. Okay, so there's your overall rate law. And in order to express this at the end of our experiment, we will need a value for each of these orders and a value for that K. All right. So here's the rub. We've only established a way of determining the order of a reactant if there's only one reactant. But we've got 2. So how do we deal with that? All right, here's how you do it. You provide in this reaction a concentration of sodium hypochlorite that is so high that at the beginning of the experiment, remember this is initial rates, kinetics by initial rates. We provide a concentration of sodium hypochlorite that's so high that at the very beginning of the reaction, that doesn't change significantly. I mean, it changes so little that it has no effective bearing on the behavior of this and that. Okay? So, in order to do that, this concentration becomes part of we call it a pseudo rate constant. Okay, that's our K. Our K prime, excuse me. So now we have a rate in terms of a constant and only one reactant. So we know how to deal with that. But you'll need to remember this part where K prime is equal to K times sodium hypochlorite to the M. All right? Because once you find out what this is, and you find out what that is, then you substitute in with this and that known, substitute in your concentration of sodium hypochlorite that I alluded to earlier. You need to determine that concentration. And then you have an equation you want unknown, and you can solve for your K. And then that K goes right here. Okay? And then the M that you determined goes there. And once you have those three values, you've got your rate law. Okay. So when we run the experiment through, we do it once at full strength at one concentration, and we get data. Now we're going to use, for this, rather than concentration, we're going to use an equivalent to concentration, which is absorbance. Okay? Now, what do I mean by absorbance? All right. I've got a couple of videos that I'm going to show you. One of them describes the use of the spectrophotometer. That is the instrument that we would be using in the lab. And fortunately, I found a video where the description is of the exact model of spectrophotometer that we would be using in the lab if we were there. And then a video to follow that with the theory behind absorbance. What does absorbance mean? Okay, so this might be a good time. Now let's hold those videos for just a few seconds, and let me review for you the way that we determine zero, first order, second order, and try to be specific. I'm going to try to be specific with the things that we're going to be, the data set that we're going to be using. So let's see. Let's say zero order. All right. So if we have the order in this column, right? We have 0, 1, 2. All right. So we have a data set of, in this case, absorbance versus time. Okay. If we plot the actual values of absorbance versus time in seconds, and we get a straight line, which would be like that, we know it's first order, right? In that case, if we plotted the natural log of absorbance versus time, okay, in that case, we would get, well, for this one, we would get a curve. And for this one, we would get a curve. Right? If it were zero order, we get a straight line here, curve there, curve there. If we plotted the natural log of A versus time, then for this one, we would get a curve. For this one, we'd get a straight line. Okay? And for this one, we'd get a curve. Okay? So that would be first order. If the absorbance in our experiment would give us a straight line here, well, actually, that's true. Right? 1 over absorbance versus time, we get a straight line. And for these others, well, actually they don't matter. But the straight line for zero order would be this data set 1 over absorbance versus time. Natural log would be first order. Second order would be inverse. Okay, so you recall from our discussion in class, that's how you determine if the rate law is 0, first, or second order for that reactant. And in this case, it's the blue dye, and we're using absorbance. Okay, okay, so I think I can erase that now. Okay. Let me see if it's appropriate now to do the pre-lab questions. I'm going to go over the pre-lab questions with you and find out where they are. Here we go. So let's look at the pre-lab questions. Okay. Number 1. Okay, the first pre-lab question says the bleach that you're using in this question is 5% sodium hypochlorite. All right. In our experiment, we're using 6%. So the calculations that we're going to do here can be used. All you have to do is take account of the difference from 5% to 6%. And you should be able to do the same calculations with the actual material that we're using in the lab. Now, as you've heard me say in class, I know, what does percent mean? Percent means parts per 100, which means it has no dimensions. The only way you can apply dimensions to percent is to know whether the numerator is mass or volume, whether the denominator is mass or volume. In that case, then you can apply dimensions to your percentage. Well, as it turns out, bleach percentage is in terms of mass, right? So it would be 5 grams of sodium hypochlorite per 100 grams of bleach. Right? That's what that means. Okay, now to the question. Well, let's see. More information we're given. The density of the bleach is 1.08. 5% bleach is 1.08 grams per milliliter. That's its density. Now, why do we need to know that? Because the question is, what's the molarity of that solution? And remember, if you've got 5% mass, how do you get to moles volume? Right? You need some sort of a conversion that will allow you to convert the mass of the bleach into milliliters or into volume, right? In order to determine the molarity. And then, of course, you have grams of sodium hypochlorite. You know how to convert that to moles, right? Molar mass. You just need the molar mass of sodium hypochlorite. You got that conversion, right? So if we're starting with 5 grams of sodium hypochlorite per 100 grams of bleach, we're going to chain our conversion factors together, right? It's one of the first topics that we discussed in general chemistry first semester. how to do unit conversions. Dimensional analysis. Okay, so what we want to do first is to convert this into volume. So if we have grams of bleach, right, 1.08, that's 1 milliliter. But molarity needs liters, right? So we need to convert that to liters. Right, 1,000 milliliters is 1 liter. There we go. Now we got our liters for molarity. We're good there. Now we need to convert grams of sodium hypochlorite. This is different grams. This is grams of bleach. The grams here is grams of sodium hypochlorite, right? So don't get those two confused. We need to convert grams of sodium hypochlorite to moles, right? So we need the molar mass of sodium hypochlorite. Well, you need to go to your periodic table, right, and find sodium, oxygen, and chlorine. And add them together because there's only one of each. And when you do that, you get a value of 74.45. Okay. Now we're able to cancel this one, then cancel this one, then cancel this one, and leaves us with moles per liter. Right, so that's our concentration of the bleach before you add it to the reaction vessel. Okay. When you add it to the reaction vessel in a certain amount, then you're actually diluting it from this amount to the actual concentration in the vessel, and we'll determine that also. All right, so if we do that calculation, you should get 0.725 molar. Okay, so if you did that with 6%, how would you change? Well, obviously that would be 6. How about density? Well, as it turns out, as you increase the amount of sodium hypochlorite in your solution, the density of the solution increases, right? So the value for density of 6% is 1.09 grams per milliliter. And that's in the method, right? So you can write this down, or you can just look in the document. It's there. Okay, so this is the answer to Prelab Question 1. All right, so let's see, Prelab Question 2. If that's the concentration of our bleach, then 2 naturally follows from it in the method. Now, since we haven't gone through the method step by step, this might be a little strange. So I'm going to try to explain it to you first. When you do the experiment, you're going to be measuring at full strength bleach. You're going to be measuring the reaction conditions as they change over time by absorbance. With the half strength, you're going to have a certain amount. First, at full strength, you're going to have a certain amount of blue dye that you add and bleach. Right? And in the half strength, you're going to add the same amount of blue dye, but you're only going to add half as much bleach. Right? So you've got to make up the other half of the volume with something. Right? So if you only use half of the bleach, what are you going to use for the other half? Right? Well, you could use water. The problem is your reaction environment is very sensitive to the ionic strength. Right? How many ions do you have in solution? So at this concentration of bleach, you can assume that it is completely dissociated. Right? It yields that and that. Okay? And these are ions in solution, right? And they affect the progress of the reaction. So if you just add water, then you really only have half of the ions that you had before. Right? So we need to balance the number of ions, the number of cations, the number of anions. The easiest way to do that without affecting the action of the bleach itself is to use something of similar strength, ionic strength, but without the reactivity. And as it turns out, table salt does that. Right? There's your sodium, and here's your anion, the chloride anion. It has no effect on the blue dye whatsoever. Why? Because there's the culprit right there. It doesn't have that oxygen. That's what's changing the blue dye. It's decoloring it. Right? So when you start out, you're going to have a certain absorbance, high absorbance, relatively speaking. And then as the reaction proceeds, the color is going to start to disappear and the absorbance value that you get from the spectrophotometer will decrease. If you just put sodium chloride in there at the same strength as the bleach, you get no reaction at all. So the only purpose here is to make sure that the ionic strength is maintained. So you put in half of this for that one, and the other half you make up with that one. Right? So how do you know that you've got the right strength? Well, if we know that the concentration of bleach here is 0.725 molar, and we put so many drops in that you're going to determine as the first stage of the experiment itself, then say you put, let's make something up, say you put 20 drops. Over here, you'll put 10 drops. So the other 10 drops are sodium chloride solution. But in order to maintain the ionic strength, the sodium chloride solution must be the same molar concentration as the bleach. Right? That's why we need to determine this first. Then we can dilute sodium chloride in a solution to the exact same molarity as the bleach. Right? So then we have a balanced ionic strength for the half-strength experiment. Okay, so let's get back to question number 2. All right, question number 2 then is, how many grams per liter of sodium chloride are required in order to prepare a solution with a concentration equivalent to the molar concentration of the bleach? Right? So our target molarity is equal to 0.725. Okay. We need 0.725 molar sodium chloride. Okay. So what does that mean? That means 0.725 moles per liter. We've got a unit conversion problem. Right? So this is where we're at. That's where we're headed. Right? Liters, don't need to change it. Moles, grams, what does that mean? Molar mass. So what's the molar mass of sodium chloride? Well, if you add up sodium from our periodic table, right? There's your sodium, there's your chlorine. You only got one of each, add them together. And you get 58.45 grams per, oops, mistake, right? No, it wasn't a mistake. Had it right the first time. Right, grams on top, grams on top. And then moles on the bottom, per mole. There we go. So moles cancel, grams per liter. And that equals to how many grams is 42.4 grams per liter to give you the same concentration as the bleach. Now, who's going to make up a liter of sodium chloride solution? I mean, that'll be enough to last me for the next 20 years of teaching this same class. Right? So we only need to make up a smaller amount. So if it takes that many grams for a liter, 1,000 milliliters, right? 1,000. How many grams would it be for, say, 100 milliliters? Right? So that's a manageable amount. Well, 100 is a tenth of that, isn't it? So we just need a tenth of the mass to be the same ratio. So that would be 4.24 grams per 100 milliliters, diluted with distilled water, of course. And that will give you the proper concentration. All right. So we got one more question to answer for the pre-lab. Okay. And that one is, this is kind of, well, not theoretical, but it's kind of reverse of the number one question. Let's see. Question is, what's the percent of sodium hypochlorite by weight for a liter of bleach? That is 0.8 molar sodium hypochlorite. So we're actually starting at the end and working backwards compared to question number one. And we're just going to assume, we have to assume, that the density is equal to 1.09 grams per mil. Okay. So this is our starting point, and what we want is a mass percent at the end. So we want whatever the percent is, mass of sodium hypochlorite. Okay. So this is another dimensional analysis. 0.8 moles per liter. We need grams of sodium hypochlorite. Right? So let's convert moles to grams. What was our molar mass? Our molar mass was 58.45. Okay, so we got that one taken care of. Now we need to convert to grams for a liter. Okay. Let's see. if we want percent, then we want grams per 100 grams, right? We want to end up with 100 grams in the denominator. So if we convert this to milliliters, right, and then we convert that to grams, milliliters to grams, right, then that's mass of sodium hypochlorite per gram of solution, but we want 100 grams. So we just have to multiply that by 100, right? It's kind of like percent. Divide one factor by the total times 100 is percent. So let me see if that works out. Takes a calculator to do it. 0.8 and 58.45 thousand, 1.09, that's 4.29, which is not the same answer that they're getting here. What did I do wrong? Pause. I made a mistake. 5845 is what? 5845 is sodium chloride, not sodium hypochlorite. There we go. You use the right molar mass. Now let's try that again. 0.8 times 74.45 divided by 1,000 divided by 1.09 times 100. Okay, so I get 5.46422%. Round it off to 2 places, and that's mass of sodium hypochlorite. There you go. When you use the wrong numbers, you get the wrong answer. Like computers, garbage in, garbage out. Okay, so those are your pre-lab questions, and they give you a place to start for determining the concentration of sodium hypochlorite in your reaction vessel. Now, there is a, I think I've incorporated, where is it? As part of the document, yes, calculation of the sodium hypochlorite concentration in the vessel. So that's part of the, maybe it's not part. Hold on a second. Let me share the screen and see if it's there. I'm going to share. Come on. There we go. Share this one. And let's look. Bleach calculations. I think this is the one. Here we go. So this gives you calculations for the actual molar concentration of bleach in your reaction vessel. And it goes through it step by step. And part of the experiment is to determine how much bleach do you need to add to the reaction mixture to get it to decolorize, that's the reaction, decolorize within 2 minutes. Right, so you go through those steps and you find out how many drops of bleach to decolorize a 5 milliliter sample of your diluted blue dye number 1. But the little container that you put into your spectrophotometer doesn't hold 5 mils. Right? It only holds 3 mils. So in this case, we're talking about here, 30 drops in 5 mils will give you that reaction. But we use a ratio to find out how many drops do you need in 3 mils, right? Because the reaction vessel in the spectrophotometer, it's called a cuvette, only holds 3 milliliters. So we need to prorate the number of drops of bleach to 3 milliliters, and that's what this step is for. So once you do that, then you can start determining the actual concentration. One other thing you do need is the mass of the bleach that you have in 30 drops. Right? That will give you the mass per drop, and then we convert with the density of 1.09 grams per mil to how many milliliters is in 1 drop. Right? Because when you add so many drops to your reaction vessel, the final concentration is different because you added a diluent. You changed the volume. Right? So you need to know exactly how much volume you added. First, you add 3 milliliters with a volumetric pipette. Then you add the bleach, so many drops, and since so many drops is that many mils per drop, then 18 drops gives you this many mils of bleach that you add to the 3 mils, and your total volume is the summation of 3 plus that value, right? So then to determine the concentration, you know how many moles of sodium hypochlorite are per drop, because, let's see, where did we determine that? Moles of sodium hypochlorite per drop. Milliliters, drops. Oh, up here. The first step is to find out how many moles per drop. Right? Then you can determine how many moles of sodium hypochlorite were added to the cuvette, and that's your concentration. And then, of course, for half concentration, all you need is to divide that by 2. So that actually does all the calculations for you. But all this needs to be reported in your formal report, right? The report has to stand alone. If somebody picks up that report and reads it, you've got to convince them that you have done these steps properly, one at a time, and your data is reliable. If you can't prove by calculation that the data is reliable and your treatment of the data is valid, then they'll just throw it in the garbage. Right? And that's probably what I should do. But since I'm your instructor, I'm gonna grade you and give you credit for some of the things that you do. But if I were a reviewer of a referee journal paper, and a paper comes in and will not stand alone and support itself as a unit, then I cover it in red ink and send it back to the author and say, fix this or don't send it back to me. Okay, so that one you've got there for your concentration of the bleach. Now, let's see, what did I, let me look at my game plan here. Done the pre-lab questions. Let's talk about safety for just a second. What are some safety issues? Well, of course, you want to protect yourself with your lab coat, eye protection, closed-toed shoes, and no open skin below the lab coat. Why? Well, some of the obvious things that you would encounter are broken glass, right? If you dropped a beaker, you dropped a test tube, that type of thing. But the other, since the blue dye is food grade, right? You don't have to worry about that. You could drink it, but you can't drink bleach, and you don't want to get bleach on you anywhere, in your eyes, on your skin, or even on your clothes. You rather hit the lab coat and let it absorb the bleach. So those are the two main issues: is broken glass and bleach. Let's see. All right, so now we can go to the procedure. All right, so let's look at the procedure. I'm going to erase this so we'll have more room. If I can get it to come off. Here we go. All right. All right, so procedure. Well, before you do the steps in the procedure, you have to gather all of your materials together. Let me unshare this thing. We don't need, yeah, let me unshare it for now. We'll come back for the spreadsheet. All right, so this is how you're gonna collect the data. All right, so in the procedure, copy of the procedure. Here we go. In the procedure, there's a page on materials. All right. So you're going to need 3 test tubes, and the test tubes are, I don't know if they're given. It's okay to just say test tubes, because the volume of the test tube doesn't matter. So you're going to need materials. 3 test tubes. You're going to need and rack, someplace to put them. You're going to need a beaker with the blue dye in it. 100 ml beaker with blue number 1, and this is diluted. Diluted dye. If we were actually in the lab, that material would already be diluted for you. The stock solution of blue dye is extremely concentrated, too concentrated to do the experiment. So we have the diluted dye, and we actually take 1 drop of the stock blue number 1 and dilute it to 50 milliliters. So that's 1 drop in 50 milliliters in this 100 milliliter beaker. Okay, so you need that for your materials. And that's what you'll draw out and dispense into your test tubes. You need 30 milliliters of 6% mass sodium hypochlorite. Okay, and you need a dropping pipette. You've seen these before. They're made out of plastic. They're all one piece. They have a bulb at the top that's squishy, and you just draw things up into that and you transfer it over. Dropping, sometimes they're called transfer pipettes. Okay. And when you do the dropping part, don't misplace that pipette because every pipette's different. They're not calibrated. So when you put so many drops in there, you better use that same number of drops to determine the mass of 30 drops. Now, I've already given that to you, but that's what you would do, and that's what you put in your report. Right? You went to the balance, you dropped out how many drops you need, and you took the mass of it so you could determine the mass of a drop. Okay, you're also going to need 2 pipettes, volumetric pipettes, 5 milliliter. You're actually going to need you need 2 of those, and then you're also going to need 3 mil. Only one of those, right? Because I said your cuvette can only hold 3 mils, so you need one that you can weigh accurately 3 milliliters. Pause the video. I'll be right back. Okay, where were we? Materials. Volumetric pipettes. You need a timer. Most of my students just use their cell phones. They have a stopwatch in their cell phones. That works fine. Once you start the timer at the beginning of your run, once you've started the reaction, you let the timer run and you read the absorbance every 10 seconds. You don't stop the timer, read, and then restart it, because that adds time in there that you're not accounting for. So once you start the timer, don't stop it till you're done. What else do we need? Graduated cylinder. Okay, 10 milliliter graduated cylinder. Of course you need distilled water. In a wash bottle. Okay. And one more thing. Oh, the most important thing. Hey. Spectrophotometer. And the one we're using is a, let's see, I think it's called a Genesis 20. Okay, and the video that I'm going to start in a minute will show you how it's operated. So we got materials. Oh, we need to do one more thing before we start the video. Determine the number of drops of bleach that's required in 5 milliliters. Right. So all you need to do for that So part 1 is number of drops of bleach per 5 milliliters to yield decolorized dye in less than 2 minutes. Okay? So how do you do that? Well, you set up your test tube rack, right? Put your tubes in it. And you, let's see, you fill one with water, right? That's as your completely no color standard. And then you have one over here with your blue dye, 5 milliliters exactly. So you use your volumetric pipette, and we learned how to use volumetric pipettes in Gen Chem 1. And you need also a blue dye here, 5 milliliters. Okay? You're going to leave that one alone. You're going to leave that one alone. And this one's the one you're going to add drops of bleach to it. Right? So if you add so many drops of bleach to that, start your timer. Timer for 2 minutes. And then you watch as this stays dark blue, this one fades, and it approaches the color of water. It'll never get there. There'll always be a faint color left, because everybody knows that bleach is kind of a yellowish color. So once the blue is gone, it's going to look sort of yellow. But it's best if your decolorization takes place, takes longer than 1.5 minutes, but less than 2 minutes, in that range. Okay, so you add it. The procedure says add 2 drops and watch it go. Right? That's not going to be enough. So what I tell my class in hands-on, you get the feel of it with your first 2 drops. Then you say, well, let's shorten this thing out. Think in terms of, well, most of my students don't know what a 35 millimeter single lens reflex camera is. That was before digital cameras. And the lens, you would be able to see through the viewfinder and through the lens. That was the nice thing about single lens reflex. And you would focus. If you were doing a manual focus, which most of them were, the high-end cameras were that, you would focus and you would go too far out of focus this way, and then you go back. So you're focusing this way, actually distance. And that's the idea behind what I'm going to tell you now. Put 10 drops in there and see if it meets the standard. It's not enough, okay? Put 20 drops in there. Does it give you decolorization in under 2 minutes? Probably not. So put 30 drops in there, right? So the idea is to go too far, decolorize in less than a minute, and then you've been putting 10 drops at a time. So let's say you went to 10 and then 40. Right? So then the logical thing would be, if 40 is too far and 20 is not enough, split the difference. Go halfway, right? Focus in. Focus too far the other way, and then you gradually get closer and closer and closer. So as it turns out, if you do this hands-on, you'll find that it takes 30 drops. In 5 milliliters of blue dye, 30 drops of bleach will give you decolorized in just under 2 minutes. So that's why the document I showed you was the mass of 30 drops, because you want to know how much bleach you actually added. And by using the density of 1.09, we can determine so many grams of 30 drops of bleach gives you a certain volume, and those calculations you'll see right there in that document. Okay, that's part one. All right, so now it's time to take a look at this video, and it'll show you how to operate the Genesis 20. It has a couple of little flaws and missing information, but it's not serious. It was the best one I could find, and it's better than my trying to draw it up here and describe it to you. You actually see somebody work with the equipment. And then after that, I'll go right into the next video on absorbance, right? The theory that allows you to measure the concentration of the blue dye as it decreases. It's directly proportional to absorbance. So as the amount of blue dye decreases, we don't have to actually measure the blue dye. We're measuring its absorbance. So they're directly proportional. You can use the absorbance as the term for your concentration of blue dye. All right, so I'm going to start those videos, and they take a few minutes, but it's worth watching. So let's see, where's my first one? This one right here.
Hello everyone, today we're going to be going over what a spectrophotometer is and the basics and how to calibrate and use it. Let's get started. This is the standard Genesis 20 spectrophotometer that you will encounter in your lab. It consists of three main parts. The display, which gives you the readout of the absorbance and the wavelength at which you are measuring. There's the interface, which you will be using to adjust the wavelength and blank the machine. And finally, the area in which you put the samples. These are the standard cuvettes that you will see when working with the spectrophotometer. Generally, you will only receive one or two. Cuvettes are designed especially for the spectrophotometer. As you can see, they have a transparent side and an opaque side. You must only hold the cuvette on the opaque side, like so. If you hold it on the transparent side, you will get oil and fingerprints, and thus you will have an inaccurate reading. Make sure you hold it on the ridged side. There may come a time when you need to adjust the wavelength on the machine. As you can see here, the machine is currently set to read at 465 nanometers. To adjust this, simply click the NM up or the NM down button. As you see here, I adjust it to 475. Whenever you begin using the machine, or after you've adjusted the wavelength, you must blank it. Simply take a cuvette with deionized water, or whichever your blank must be, take a Kim wipe, and gently wipe the transparent sides of the cuvette. Be careful not to scratch the sides of the cuvette, as that will produce inaccurate readings. Next, open the hood and firmly and carefully insert the cuvette, making sure not to spill any drops into the machine, and close the door. If you look closely, you will see an arrow alongside where you put in the cuvette. That is the way that the light passes, therefore your transparent edges must be facing the same way. Put the cuvette in, and again make sure that the light passes through the transparent edges alongside with the arrow. Now you're ready to blank the instrument. Simply hit the 0 abs button as you see there, and it will set the blank. Once the instrument readout is set to 0, you know that you're ready to go. Without adjusting any settings, simply take out your blank, holding the opaque side of course, and insert your cuvette with the solution of which you'd like to take the absorbance. Make sure that you take a Kim wipe and wipe the transparent sides of the cuvette to make sure that there are no smudges or drops of solution on the side. And again, place it in the machine, facing the correct way, and close the door. Once you've inserted the cuvette with your sample, simply close the door. Do not adjust any settings. All you need to do is wait for the reading to stabilize. The reading, as you can see, is right there. Here it finally stabilizes at 0.107. Remember, absorbance does not have units. And there you have it, the basics of a spectrophotometer. Good luck and have fun!
Okay, now let's go to the next one. So when they were pointing to the NM, that stands for nanometers, right? So that's the wavelength at which the instrument is reading the absorbance. Right? Your wavelength is going to be 630 nanometers, and that's in the method, and I've written it on the whiteboard here for further discussion. The ABS, the ABS, is an abbreviation for absorbance. Right? So when you set the machine to 0, or you blank the machine with your distilled water, you're telling the machine that this container, with whatever it contains, in this case water, is your 0 point. No absorbance. Right? So even if it does have absorbance, you're telling the machine to set it at 0 there. Okay, so let's go to the next. Here we go. And this is going to be a theoretical discussion, and this is from Khan Academy. So many of you have been to Khan Academy, and you'll recognize the voice of the founder of Khan Academy speaking.
What I want to do in this video is to talk a little bit about spectrophotometry. Photometry, which sounds fairly sophisticated, but it's really based on a fairly simple principle. So if I have, let's say we have two solutions that contain some type of solute. So that is solution 1, and then this is solution 2. And let's just assume that our beakers have the same width. Now let's say solution, let me put it right here, number 1 and number 2. Now let's say that solution 1 has less of the solute in it. So let me make, so that's the water line right there. So this guy has less of it. Let's say it's yellow, or to our eyes it looks yellow. So this has less of it. So this has, actually let me do it this way. Let me shade it in like this. So it has less of it. And let's say solution number 2 has more of the solute. So it's more. So I'll just kind of represent that as more closely packed lines. So the concentration of the solute is higher here. So let me write higher concentration. Higher concentration. And let's say, and this is a lower concentration. Now let's think about what will happen if we shine some light through each of these beakers. And let's just assume that we are shining at a wavelength of light that is specific to the that is specifically sensitive to the solute that we have dissolved in here. But I'll just leave that pretty general right now. So let's say I have some light here of some intensity. So let's just call that the incident intensity. I'll just say that it's I0. So it's some intensity. What's going to happen as the light exits the other side of this beaker right here? Well, some of it is going to be absorbed. Some of this light at certain frequencies is going to be absorbed by our little molecules inside the beaker. And so you're actually going to have less light coming out from the other side, especially less of those specific frequencies that these molecules in here like to absorb. So you're going to have less light come out the other side. So you're going to have less light come out the other side. I'll call this I1. Now, in this situation, if we shine the same amount of light, So I0, if we shine the same, that's supposed to be an arrow there, but my arrow is kind of degrading. If we shined the same amount of light into this beaker, so it's the same number, that and that is the same intensity of light, what's going to happen? Well, more of those specific frequencies of light are going to be absorbed as the light travels through this beaker. It's just going to bump into more molecules because it's a higher concentration here. So the light that comes out, when you have a higher concentration, I'll call that the intensity, I'll call that I2. This is going to have a lower intensity of light that's being transmitted than this one over here. In this case, I2 is going to have a lower intensity, is going to be less than I1. And hopefully that makes intuitive sense. These light, if you imagine photons, are just going to bump into more molecules. They're going to be absorbed by more molecules, so there will be fewer that make it through than these right here, because here it is less concentrated. It's also the case if the beaker was thicker. So if you have another situation, let me draw another beaker. If you have another beaker that is maybe twice as wide, it's twice as wide, and let's say it has the same concentration as number 2. We'll call this one number 3. We'll call this one number 3. It has the same concentration as number 2. So I'll try to make it look fairly similar than this. Fairly similar to this. And you were to shine some light in here. Some light in here. And in general, you want to focus on the frequencies that this is the best at absorbing. But let's say you shine the same light in here, and you have some light that makes it through, that exits. And this is actually what your eyes would see. So this is I3 right there. What do you think is going to happen? Well, it's the same concentration, but this light has to travel a further distance to that concentration. So once again, it's going to bump into more molecules, and less and more of it will be absorbed. And so less light will be transmitted. So I2 is less than I1, and I3 is actually going to be the least. And if you were looking at these, this has the least light. This has a little bit more light being transmitted. This has the most light being transmitted. So if you were to look at this, if you placed your eyeball right here, so if you placed your eyeball right here, those are your eyelashes. This one right here would have the lightest color. You're getting the most light into your eye. This would be a slightly darker color, and this would be the darkest color. That makes complete sense. If you dissolve something, if you dissolve a little bit of something in water, it'll still be pretty transparent. If you dissolve a lot of something in water, it'll be more opaque. And if the cup that you're dissolving in, or the beaker that you're in, gets even longer, it'll get even more opaque. So hopefully that gives you the intuition behind spectrophotometry. And so the next question is, well, what is it even good for? Why would I even care? Well, you could actually use this information. You could see how much light is transmitted versus how much you put in to actually figure out the concentration of a solution. That's why we're even talking about it in a chemistry context. So before we do that, and I'll show you an example of that in the next video, let me just define some really some terms of ways of measuring how concentrated this is, or ways of measuring how much light is transmitted versus how much was put in. So the first thing I will define is transmittance. Transmittance. And so when the people who defined it said, well, what we care about is how much is transmitted versus how much went in. So let's just define transmittance as that ratio. As that ratio. The amount that gets through, so in this example, the transmittance of number 1 would be the amount that got through, over the amount that you put in. Over here, the transmittance would be the amount that you got out over the amount that you put in. And as we see, this one right here will be a lower number. I2 is lower than I1. So this will have a lower transmittance than number 1. So let's call this transmittance 2. This is transmittance 1. And transmittance 3 is the light that comes out, that gets through, over the light that goes in. And this is the smallest number, followed by that, followed by that. So this will have the least transmittance. It's the most opaque, followed by that, followed by that. Now, another definition, which is really kind of a derivative of the not in the calculus sense, it's just derived from transmittance, and we'll see it has pretty neat properties, is the notion of absorbance. Absorbance. And so here we're trying to measure how good is it at absorbing. This is measuring how good are you at transmitting. A higher number says you're transmitting a lot. But absorbance is how good you're absorbing. So it's kind of the opposite. If you're good at transmitting, that means you're bad at absorbing. You don't have a lot to absorb. If you're good at absorbing, that means you're not transmitting much. So absorbance. Absorbance right here. And that is defined, absorbance is defined as the negative log of transmittance. Or you could view that, and this logarithm is base 10. Or you could view that, if the transmittance we've already defined, as the negative log of the light that is transmitted, is transmitted over the light that is input. But the easiest way is the negative log of the transmittance. So if transmittance is a large number, absorbance is a small number, which makes sense. If you're transmitting a lot of light, the absorbance number is going to be very small, which means you're not absorbing that much. If transmittance is a low number, that means you're absorbing a lot. And so this will actually be a large number. And that's what the negative log gives us. Now, what's also cool about this is there's something called the Beer-Lambert law, which you could verify. And we'll actually use this in the next video. The Beer-Lambert law. I actually don't know the history of where it came from, and I'm sure it's based on somebody named Beer, but I always imagine it's based on someone transmitting light through beer. The Beer-Lambert law, and this tells us that the absorbance is proportional. I should write it like this. The absorbance is proportional to the path length. So this would be how far does the light have to go through the solution. So it's proportional to the path length times the concentration. Usually we use molarity for the concentration. Or another way to say it is that the absorbance is equal to some constant. It's usually a lowercase epsilon like that. Some constant, and this is dependent on the solution or the solute in question, what we actually have in here, and the temperature and the pressure and all of that. It's equal to some constant times the length it has to travel times the concentration. Let me make it clear right here. This thing right here is concentration. And the reason why this is super useful, is you can imagine, if you have something of a known concentration. Let me draw it right here. So let's say we have an axis right here. That's axis, and over here I'm measuring concentration. This is our concentration axis. And we're measuring it as molarity. And let's say the molarity starts at 0. It goes, I don't know, 0.1, 0.2, 0.3, so on and so forth. And over here, you are measuring absorbance. In the vertical axis, you measure absorbance. Just like that. Now, let's say you have some solution, and you know the concentration. You know it is a 0.1 molar concentration. So let me write down M for molar. And you measure its absorbance, and you just get some number here. So you measure its absorbance, and you get its absorbance. So this is a low concentration, didn't absorb that much. You get it at, I don't know, some number here. So let's say it's 0.25. And then let's say that you then take another known concentration at, say, 0.2 molar, and you say that, oh, look, it has an absorbance of 0.5. So let me do that in a different color. It has an absorbance right here at 0.5. And I should put a 0 in front of these. 0.5 and 0.25. What this tells you, this is a linear relationship. That for any concentration, the absorbance is going to be on a line. And if you want a little review of algebra, this epsilon is actually going to be the slope of that line well, actually, the epsilon times the length will be the slope. Well, I don't want to confuse you too much. But the important thing to realize is that you have a line here. you have a line here. And why? And the reason that's useful is you could use a little bit of algebra to figure out the equation of the line, or you could just look at it graphically and say, okay, I had two known concentrations, and I was able to figure out the absorbance because I know, well, I just measured those, but because I know that it's a linear relationship, the Beer-Lambert law, and if you just kept taking measurements, they would all show up along this line. You can then go the other way around. You could then measure for some unknown concentration. You could figure out its absorbance. So let's say there's some unknown concentration, and you figure out its absorbance is right over here. Let's say it's 0.4. It has an absorbance of 0.4. Then you can just go on this line right here, and you say, okay, well, then that must be a concentration of this, well, whatever number this is. And you could measure it, or you could actually figure it out algebraically. And so this will be pretty close to 0.2 molar, a little bit less than 0.2 molar. And we're going to actually do an example of that in the next video.
It's a pretty good explanation of how to operate the Genesis, which I misspelled earlier, the Genesis 20. And then what does absorbance mean? And it's really important, the Beer-Lambert law, which says absorbance is equal to that value times that value times that value. This is the concentration, right? This is the path length. How far does the light have to travel through the solution? And this is what's called the molar absorptivity. It is characteristic of the molecule that is absorbing the light, in this case, the blue dye number 1, right? So since if we put a cuvette into our instrument, the path length doesn't change. It's always 1 centimeter. So that's constant, and that's characteristic of the molecule. It's constant. So these two are constant. So that means that if we know the absorbance, then the relationship is linear, and if the concentration decreases, the absorbance decreases. That's why we can use absorbance to represent our blue dye concentration. Okay? All right. So what wavelength are we going to use? 630 nanometers. So instead of 473, I think it was, on the Genesis 20, you hit the up key for nanometers, and you go all the way up to 630. Let it sit there and warm up at that wavelength. So it's stable. Then you go through the procedures. All right, so what would the procedure be for your experiment? Right, we're going to do this part 2, I think, is for full strength bleach, and I put it in quotes because it's not really full strength, because that would be 6%. It's been diluted. Since 30 drops of 6% bleach in 5 milliliters gives you the right timing for your experiment, then we prorated that for 3 milliliters, and it turns out 18 drops is for 3 milliliters, right? So how do you actually conduct the experiment and gather the data? This is where the data comes from. You have your cuvette, right? And the light is going to be traveling the way Kahn put it, the incident radiation, and then it's going to produce some output here, right? So this will be the opaque side, and then the transparent side is on either end, and that's the one that you need to clean with your Chem wipes, because you don't want any fingerprints on it. That's why you probably want to wear gloves. One reason, of course, is bleach. You don't want to get bleach on your hands, and you don't want to get fingerprints on your cuvette, right? So you're going to put 3 milliliters in here, exactly 3 milliliters, right, of the blue dye solution that's been diluted for you. And then you're going to, well, first you're going to put 3 milliliters of water and blank the instrument. And once you blank the instrument with water, then you pour the water out of your cuvette and use the exact same cuvette, because they're a little bit different from one to the other. So you don't want to blank it with a different cuvette. Blank it with this one. And then you pour the water out, shake it out real good. You could even cram a Chem wipe down in there and absorb all the droplets. And then you put 3 milliliters of your blue, number 1. Okay? Now, then you put the cuvette in the spectrometer. After you blank it, put the blue dye in the cuvette in the spectrometer, properly oriented. And then you put 18 drops of dye in there while it's in the instrument. So be careful and don't spill any bleach in the spectrophotometer. Why are we doing it that way in the spectrophotometer? Because this reaction is fast. The minute that bleach hits the blue dye, it's going to start reacting. Right? So you're going to miss the first few seconds of the reaction as you add the 18 drops. But once the 18 drops is in there, then you want to use a little, maybe a little stir rod, put it in there and mix it around. Right? Don't take too long, just mix it, and then close the lid. Start your timer immediately. And as you immediately start the timer, somebody else in your group will read the absorbance. Right? And the absorbance might be something in the neighborhood of 0.4. I'm just spitballing here. 0.4 absorbance or something like that. And then after the next 10 seconds, whoever's got the timer says time, and then the other person reads the absorbance, reads it out, and then the third person of your group records it, right? And you might, every 10 seconds, so this is time 0 in seconds, and then 10 seconds you might get 0.4, no, not 4, maybe 3, 8, and maybe 3, 6 for the next one, and it just keeps going. And the method says keep doing that until you reach a less than 0.1, I believe. I think that was it. Less than 0.1. You can go lower than that. In fact, the data set is already there for you. So you just take what the data set gives you. I think we went down to maybe 0.02, something like that. So you have a complete data set there for that. And that's it. Those are the numbers you have. Time versus absorbance, right, for the full strength. Then you go back, part 3, right? And this one you're going to do at half strength. All right? So what does that mean? You need half as much bleach, right? So instead of 18 drops, you have 9 drops. And 3 milliliters plus 9 drops of sodium chloride solution, right? And the concentration of that is, well, let's see, I didn't give it to you before, but in fact, I did leave that one out. So your materials, add this one to your materials. 0.878 molar sodium chloride. All right, so how do we do this one? Well, you want your bleach to be the very last thing you add, right? Because I said the reaction is going to start immediately. So you add your 3 mils in here, then you add 9 drops of the sodium chloride solution. And in order to do that, you want to use the same dropper, but you want to clean out the dropper. Right? So use distilled water in a beaker, clean out your dropper, squirt it out real good. And then to be sure that you haven't diluted your sodium chloride solution, just draw up some of this solution and squirt it out into your waste beaker. Right? Clean out and just be sure that you start with that much in there. Then you pull it up and add 9 drops. Right? Then you take that same dropper, squirt it out real good, draw up some of your bleach, squirt into your waste beaker, right, as you clean out your dropper. Then you draw it up and put 9 drops in and start your readings immediately. Now at half strength, the decolorization is going to proceed at a much slower rate. Right? So now it's going to take more than 2 minutes. Right? So you're going to have a longer data set, and you can see that in the document that I've given you in Blackboard, that you'll have a short data set for the full strength and a longer data set for the half strength. So that's all your data. That's it. You got it. You just need to clean up, rinse out your cuvette, turn it upside down on a chem wipe and let it dry out naturally. You can even turn the spectrophotometer off if you want. And then do your calculations. Okay? So now I'm going to show you what to do with your data. Now I'm going to show you how to use your data in Excel to take some of the drudgery out of the process and produce a more refined output. So to do that, I want to share the Blackboard, and let's see. It's going to be right here. So there's the Chemical Kinetics lab with the documents that are there at present. And here's your sample Excel spreadsheet. All right. So now I'm going to share the spreadsheet itself that you will have to download in order to access. And this is a bunch of stuff in my way. You can't see it, but I can. All right. So this spreadsheet has been completed with a data set for a different type of experiment. Well, it's the same type of experiment. It's a different dye. Instead of using blue number 1, this experiment uses red number 40, which just means that you have a different wavelength here, 485 nanometers instead of 630. And the kinetics are going to be different. But the process for manipulating the data is the same. So the full strength data set is over here, and instead of 18 drops in, or 30 drops in 5 mils, this would use 8 drops in 5 mils, and would consequently have to be prorated for 3 milliliters in the cuvette reaction vessel. So your data set here is for time, it's your x-axis, and the first set you would graph is the straight concentration data in the form of absorbance. So you just lay it out like that. Each time interval has an absorbance, and then you want to transform each one of those, because if you graph time against absorbance or concentration, and you get a straight line, that's 0 order. If you transform that data into the natural log here and graph x-axis time and y-axis natural log, You get a straight line there, that's first order. And similarly, the reciprocal graphed as a straight line would be second order. So you've got to graph each one of these to determine which one gives you the proper order for the dye reactant. All right, so how do you do that? Well, you have to put this data in by hand, right? So you can see up here, each one of those numbers is put in by hand, and then we just format it to make it look nice. Click, hold, and highlight that group, right click, gives you the format the cells. Then you say number, how many decimal places, and that's why you get 3 decimal places for everything in that column. Now, how do you make the transformations? Well, in Excel, you know that any formula starts with an equal sign. So here we have the equal sign, and then LN is the formula for natural log, and then within parentheses, your reference cell. And the reference cell for this one is just next door, B5. Right? So once you've got that one input, then the rest of them are already there now, but for you to do it, just go to the lower right-hand corner of that cell and grab that handle, click, hold, and drag it down. And it fills all the other cells with the natural log of the relative cell to the one that you're referencing here. This one would be B17. There it is right there. Okay. Do the same thing for the reciprocal. So it's equal 1 divided by then the reference cell, which would be B5. Right. So once you've got that input, you just grab the handle, drag it down, and it fills those. So now you have all your data transformed for you without the drudgery of doing it by hand. And to put this data set as is into your Word document for your report, you would just highlight all this information that you want to put in there, and then copy. Just right click and copy, right? And then go to your document and paste it wherever you want. It will paste it as an Excel probably input, but it will still be formatted the same way. If you want to format it differently, then you would have to export it as something else. You might even be able to export. That's going to waste too much time looking that one up. Okay, so now you have all your data, and you need to periodically save, of course, right? So you don't lose all your work. Now, how do you graph? Well, to produce a graph of this column as your x-axis and its neighboring column, and I'm clicking and holding as I drag, and I've identified this column is X, this column is Y. That's default in Excel. It assumes that the first one to the left is your X axis and the next one to the right is the Y axis. Now that you've got them highlighted, you just say Insert and go to Charts and pick out the scatter plot. Right, click that little arrow to the right, and then pick out either the pure scatter plot or the line connected. That's fine. Do it like that. So now you've got your plot, and it looks exactly like this one because it's using the same data set. Then what you want to do is change the title to be more descriptive, of course. And then you want to add, over here, this plus sign, you want to add an element which would be axis titles. Right? And that gives you titles for your axes. And you change those to be appropriate for the axis. Now, if you want a trend line, we're looking for straight line, right? So if you click this trend line to the right and say more options, then you get more than just linear. Linear is default. And then you can say, you can give the equation for the linear line, right? And you can give the R square, and I'll describe R square in a second, what it means. There you go. So in that case, you have the line with its slope and its y-intercept for the pure data. And you'll notice that the trend line, straight line, is nothing near this curve. That curve, and you can prove that mathematically with the R square value. R square value is a way of expressing statistically how good the fit is for a particular formula. So this line is a straight line, so it draws a straight line, and then it says, how well does that data fit the straight line? Well, 0.8 is, sometimes that's considered good, but in our case, we can see that it's not. A perfect fit would be 1.000. All right, so let me leave that one. I'll just leave that one here for now. I'm not going to save it, but it's good for descriptive purposes. Now, how do you do the next one? The next one is going to be, well, let me get this out of the way. Next one's going to be the natural log. Right? And, okay, the natural log, we're actually using the wrong, see, I've got the wrong wavelength input here. See, so that's an improper description. That should be absorbance at 435 nanometers, 485 nanometers, I think. Yeah, there. So that's an improper description. I need to change that. So how do we do the natural log? Well, you can't just drag across like this, like that, because then you've got the absorbance and the natural log of the absorbance. So what you do is you click and hold and highlight the x-axis and release it, and then go over here and hold down the control button and click and hold and drag down for your y-axis. So now you have your two axes identified, and you just go ahead and do the same thing we did for the first one in the insert and pick out the scatter plot with connected lines. There we go. Right. So there is our natural log plot. And you do the same type of changes for this one. You get a trend line, and the trend line looks a whole lot better. It's closer. And if we go in and as we add the trend line, right, we'll be sure that we're selecting more options. Then we can select display the equation and display the R square value. And here they are, right down here. Put them in a more visible place like that. And you notice here the R square value is 0.995. That's very good. And you can see from the drawing of the trend line, that small dotted line, that it very closely fits that value. So we suspect that it's first order. In order to prove that, of course, you need to go and finish your plots with the inverse plot. And you just do it the same way. Take the data, highlight the X set of data, then hold the control key and drag down on the Y set. Right? And then you have those identified, tell it to graph it, and then you have your three plots side by side, and you can prove that this one in the middle, first order, is the proper one. Now, notice here that this plot, by default, Excel puts the axis labels, right, next to the 0 mark. And since we have negative numbers here, we have our x-axis labeled in the middle of the graph. That doesn't look very good. So what we need to do is highlight the graph, and actually we want to go in and highlight the y-axis. Then we want to right-click it and format the axis. And when you format the axis, you go down and select where does it, yeah, hold on a second. Horizontal axis crosses. Where does the horizontal axis cross the vertical axis? Right, we highlighted the vertical axis. Now we're going to tell it, instead of automatic, we're going to give it an actual value. And notice from the graph that the bottom number on the y-axis is minus 3.5. So you axis value here, you put minus 3.5 and click anywhere else. Click back out in the, there we go. And it moves the axis labels down here on the bottom. Now that looks better. Okay, so the other things you need to do, of course, label the axes, change the descriptive up here, and then you have, once you do that for all three of them, you've got those available to cut and paste into your Word document for your report. Okay, so we've identified the formula for this straight line, y equals mx plus b. And remember when we discussed the integrated rate laws. The integrated rate law as a straight line had to be either raw data, natural log, or inverse. And for this one, since we know it's a straight line, that's the one we want to focus on. All right, so we want to know what is the k value. Remember, natural log equals minus k minus k times t time plus the natural log of the zero value for a, which is the y-intercept. So focusing on the slope of the line, in this place right here, we know that the k value is 0.0467. Okay? So that way you have the, actually this is the k prime value, right? Because we did a transformation to limit to one reactant as the changing value. So 0.0467 is the k prime value for full strength. Right? Now you can get fancy and you can generate a summary statistics here. And remember, the first year general chemistry, the first lab we did was how to do that right here. How to do that in Excel. I can show you really quick. Actually, it's going to take too much time to do that. So in order to do that, what I'll do is I'll make available the first lab from the Gen Chem 1, and I'll put it in the lab number 2. So I haven't done it yet, but I will do it. And you can have access to that information that you may have misplaced over the intervening months. But when you do that, I inserted these equations here just to illustrate the fact that we're talking about a first order equation, which means that the slope of the line is minus k prime. Right? So there is, this is your actual value here. is 0.0467. And of course, talking about a disappearance of something, that's why the negative value. But we tend to write the law as a positive value, and the change in the dye takes care of the negative. Okay, so you've got that value. You do the exact same thing for your half strength data set. Notice the half strength data set is larger because it takes longer at half strength concentration of bleach for the dye to be decolorized. And in this case, we're talking, but the same thing happens with the blue dye, only at a different kinetic rate. So we generate the 3 plot, and we find that, yes, indeed, even at half strength, we still get a linear plot for the natural log of the concentrations. So we have that one fixed, and we, when we created the trend line, we told it to also put the formula and the square value in there. So now we have 0.0275 as the K prime for the half strength. Notice the K prime is different for the full strength and the half strength, and that's important. We're going to use that information in a few minutes to deduce the order for the bleach. All right, now I'll show you how to do that. Okay. So that being said, that's all the information we need from this plot of this information, the data that we're working with. All you have to do is create a similar spreadsheet with the data that's provided in Blackboard for you for the blue dye, and just parrot what I've done here, and you'll get the information you need to proceed to the next step. All right, so I'm going to unshare this, and we'll continue with that in just a second. Let's see. Stop share. Okay, so the rate law for the blue dye, as well, for the red dye as given in that data set. So in this case, the one that we're dealing with right now number 40 is first order. Okay? Now you'll need to do that with your blue data set and prove what order it happens to be, whether it's 0, 1, or 2. Right? If it's 0 in, then that term disappears. Right? Anything to the 0 power is 1. Okay, so we've determined the order for our red number 40, full strength bleach. All right, so let's go back. Let me share that again. Here we go. Share. And let's minimize that. There we go. Now, this actually gives you all the information you need, because what is if our order is first order, then we know that the integrated rate law for that one is right down here. Natural log of A equals minus K prime T plus natural log of A0, our starting concentration. Right? So the slope of the line is a negative K prime. So the slope of the line in this case is 0.0467 minus 0.0467. Okay? If that's the negative K, then the K prime is equal to 0.0467. Right? So that goes right there. Well, no, you can't see that. That's a sad state of affairs. But the K prime is there in the equation. You just have to get rid of the negative sign, and you have the K prime for full strength, right? So K prime full strength. Let me use my little device. Right. So here it is. Oh, no, that's not it. Let's not let me. It's making me select. Okay, that's not good. So it's explained over here. Here we go. So the natural log of the A concentration is minus 0.0467 times time plus the y-intercept, which means the rate law for that one in terms of K prime is 0.04671. Right, that's the K prime. And we got a negative there because it indicates that the dye is disappearing. But the K value itself is 0.04671. Now we do the same experiment with half strength, and notice we get a bigger data set. So we just plot the data, and this data is not quite as good, but it does tell you that the reaction is indeed first order, right? Because you get the same result for the half strength, but you get a different K prime for half strength, right? Now the K prime is 0.02747. All right. So those are two different K primes. Why is that? Well, let me unshare that. Stop share. Okay. So if the first K prime is 0.0467, right, and the rate law was originally actual K times sodium hypochlorite to the M, and then in this case red to the first power. We already discovered that one, first power. Then what's the K prime? Well, the K prime is equal to K times sodium hypochlorite to the M. Right? Well, we've got two different K primes. We've got this one for full strength, and then we've got K prime for half strength as 0.02. Shoot, my memory's not that good. Let me get over here. 2747. Okay, let me go back. Where am I? Here we go. 2747. Right, for half strength. All right. So these are two different values, and this full is like that, and this half prime then would be the same K. That K didn't change. So what changed between these two? Well, these two changed. Those stayed the same. These two stayed the same. The concentrations is different, right? In the full strength, we have a concentration that we determined, right? We calculated it. And then for half strength, it's a different concentration, right? So what is that? Well, let me see. What is the concentration? Hold on a second. Let me look. See if it's in. I'm not sharing this. I'm just going and looking for it. It's not there. Here we go. All right. 3.919, 919. Okay, so you could probably still see me because I didn't share that screen. The concentration in this one is 3.919 times 10 to the minus 2. 3.919 times 10 to the minus 2 molar. And the concentration in this one is 1.9601. 1.9601 times 10 to the minus 2 molar. All right, and the K is unknown, but we do know the values for this. All right, so let me get back to where this. So there's the concentration of full strength, there's the concentration of, and this is actually in the reaction vessel, in the cuvette. So what are we going to do? Well, we're going to say the K prime is this value, so we're substituting here 0.04667, and this one is 0.02747, right there, for that value, this value, and this value. Right, so we have these two equations. The problem is they're two unknowns, this one and that one. But that's really not such a problem, and I'll show you how that's not a problem. But I have to erase some things to get there. All right, so let me erase this. So here's what you do. Once you've determined these concentrations and the K prime for each one, right, and you did that from your plots, then you ratio them. You divide one by the other. So if we divide this one, 0.0467 divided by 0.02747 equals this one divided by that one. K times this concentration to the M, and this one, this concentration. So 1 times 10 to the minus 2 to the M. Okay? Now, notice that these two K's cancel out. So now we only have to deal with the M, right? We've already got the N, which is 1. That's for the dye. And since this is full strength and this is half strength, you know that the ratio between these two is 2, right? Divide that into that, you'll get 2. So how do we resolve this expression? Well, that's this to that power and this to that power is the same as this one divided by that one, all to the M power, right? So this one divided by that one is in here, and this is equal to 2, right? So 2 to the M power is equal to this, okay? So what is this one? That's the one where you have to just do the calculation. So 0.0467 divided by 0.02747 is 1.7. So this is equal to 1.7. All right. That's not really a problem, but it requires that you would do some logarithmic calculations in order to find out what M is, right? You've got an equation in one unknown, you just need to solve for it. So I can show you how to do that. There are two ways. There's the rough estimate way, and then there's the exact way. The rough estimate way is you say, all right, we want whole numbers. All right, let's just say we want as near to whole numbers as possible. So what's the closest whole number to 1.7? Is 2. Right? So if we say that's approximately 2 is equal to 2 to the m power, what's m? Right? M has to be 1. Right? 2 to the first power is 2. So in that case, m equals 1. Okay? So it works better if your ratio here is closer to 2. If it's like 1.95, sure, call it 2. But in this case, if we need to find out exactly what m is, and remember, I said in the lecture that the orders don't have to be whole numbers. We studied them as whole numbers because it's easier to deal with the information and get a handle on what's what. So in calculating m that I'm going to do next, the value won't be a whole number. Right? So that's what you would do if you're stuck with a value that's not close enough to round to a whole number. So what do we do? Well, if we've got 1.7 equals 2 to the m power, how do we find m? The easiest way is to use logarithms. And I'm talking about common logarithms. So if we say, take the log of both sides. So the log of 1.7 equals m times the log of 2. Right? The power becomes a multiplier. Then you say m is equal to the log of 1.7 divided by the log of 2, and just resolve that. Right? I'm sure there's a transformation that we could use to combine those logs before we do it, but it's simpler just do the log of each one. So 1.7, take the log of that one, is 2.304 times 10 to the minus 1. And the log of 2 is 3.010 times 10 to the minus 1. And divide those two, and you get 0.77. Right? So m equals 0.77. It's something less than 1. So this is exact, this is approximate. So just so you don't panic, you won't have to do this calculation with your data set. I have set it up and massaged the data so that this one will work for you. Okay? This is what you would do if I were nasty and I just left the data raw and you'd have to deal with it. But I changed it around so that it would give you this one. Okay, so now that we know the m and the n for this reaction, we can calculate the k. Okay, how do you do that? You go back to the original assumption. Remember what we said? K prime is equal to K times the concentration of sodium hypochlorite to the M, which we're going to say is 1 for simplicity's sake, rather than 0.77. So, what's the K prime? Well, You could pick one of either this one or that one. It doesn't matter which, because the K prime is this value for that one, and the concentration that goes with it is that one, right? Okay. So let's use the first one. 0.0467 equals K, which is our unknown, and then the concentration of 3.919 times 10 to the minus 2 molar to the first power. Right? So now we have an equation and one unknown. Solve it. So that means since this is to the first power, it's just that number, and K then is equal to this one divided by this one. Right? So let me do that little calculator trick. 0.0467, 3.919, that's 2 divide. And our K then is 1.19. Okay, there you go. This is for the red dye data. You just need to duplicate these steps for your blue dye data, and you're home free. But we need to write the rate law for the entire equation. Right? So now what we need to do is go back and say, for our red dye, the original rate law was this, would be K times the concentration of sodium hypochlorite to the M times red to the N. Right? So now we say K is 1.19. I think that's right. Yeah, 1.19. And then whatever the concentration of this happens to be to the first power, and this one to the first power. That's the rate law right there. Okay? That we did all of this mess we've done to arrive at that one simple equation. All right. So let me see. We've got that. We've got that. Calculations, order. Okay. So all you have to deal with now is the post-lab questions, right? And I'm not going to give those to you. Right? I did the pre-labs for you. Post-labs, let's just go through them and I'll see if there are any strange tricks in here that I can help you with. Post-lab question 1. The data in this experiment obeys the Beer-Lambert law. Explain this law, right? So you saw the Khan Academy video that explains the Beer-Lambert law. Just put it in your own words. Number 2. Why is equimolar sodium chloride solution used to dilute sodium hypochlorite in the second trial, in the half-strength trial, instead of distilled water? I already gave you that answer, right? So if you don't remember, go back and look at it in the video. Number 3, sketch the graphs of concentration versus time for a single reactant in which, let's see, it's better with formulas on a whiteboard. And they're saying this one yields products. For that one, if it were zero order, or if it were first order, or if it was second order, right? So what does that mean? Well, I'll give you a hint. Every graph that you draw, zero, first, second order, is going to be a straight line. The trick is what's on the axis. What's the transformation? So you just sketch those out. Okay, you can do them freehand, or if you have a paint program or something where you can do it on your computer and then print it out and paste it, or copy and paste into your document, that's fine. Or just do it by hand and photograph it and plug that in. Okay, number 4. What does number 4 say? Write a general equation and rate law for a reaction having 2 reactants. Well, that's simple. We've done one. If you have something that's, let's see, I'll give you the equation. Let's say we have so much A, so much of B yields products. Write the rate law for that. Right. Now, I'll give you a hint. Can you use these values as powers in your rate law? This is an overall equation. The answer is no. The powers have to be some other. Don't use A, don't use B. Okay? You know how to write the rate law. Now, number 5 says, write the rate law for a reaction having 2 reactants in which one is in excess. That's what we did for this experiment. One was in excess, which means bleach, right? But we want to use this format. Make one of them excess. Then what do? Right? Well, you can't use K now. Well, you can use K, but you have to make the transformation, right? And define what K prime is, then write that as a rate law. Okay, that's your hint. Okay, number 6. I think this is the last one. Yeah, number 6. The orders with respect to reactants C and D of a reaction were determined experimentally to be 2 and 3. All right? So if this were C and D, if this were like C and D, then write the rate law, right, with the orders for C, the order for D, in the rate law. You know how to do that. Then the question is, what's the overall order for the reaction? You remember what that is? The overall order for the reaction is just the sum of the individual component reaction orders. Right? So if order's 1 for one and 1 for the other, then the total is 2. Now, part B of 6 is the tricky one. What would happen to the rate of reaction if only the concentration of C were doubled? Right. So, if the order of C is 2, and you double C to where it's twice what it is here, what happens to the rate? Well, if you double C, it's got a 2 in front of it now, right? Well, everything inside that bracket has to be squared. So what happens to the rate? If you square 2, what do you get? 4. So you increase the rate by 4 times if the order is 2. If the order is 3 and you double it, then it's 2 times 2 times 2 is 8. Okay, that's your hint. If you do it to both components, then you apply the order to each of the increases, right? 2 times, 3 times, half, whatever it happens to be, and then you multiply them together, right? Because that is the effect on the rate. Right? That's all the hint I can give you without just telling you the answer. Okay. So I think we've come to the end. Yes. So the only thing that I haven't given you any hints on for your formal report is, what do you put in the summary and conclusions? Well, a summary is just a summary of what happened and the results of the experiment. So you could summarize in terms of what's the rate law. That would be a good way to summarize it. Rate law was determined to be blah. But conclusions requires that you have to think. And the rubric has 2 or 3 categories that apply to that. One of them asks you for application to real life situation. How would knowing the rate of a reaction and the effect of changing concentrations, how would that be applicable in real life? That takes some thinking. You might need to do some research. And interpreting the data, conclusions, you definitely do want to include a blurb on where are the possible sources of error. And if we repeated the experiment, what changes would I make to minimize those errors? Or even if you just wanted to investigate the experiment a different way, right? Say instead of half strength, you wanted to do quarter strength or third strength. Things like that. This is where you show whether you're thinking or not. The summary and conclusions is really what a reader of your research paper would want to see. They of course want to know that your data is solid, and they've already determined that. Now they're going to the conclusions, and this is what you think the results mean, right? What effect do they actually have on the overall understanding of kinetics? And what would be the effect of changing certain parameters? What would be those effects on the kinetics of the experiment? Okay, so that's where the thinking comes in. And that's where we separate the sheep from the goats. Okay, that's it. I know this is going to be a long video, but I had to put in all that detail so that you would be able to write the report with some competence. And good luck.